Good morning, student. Now we'll study about next part from this unit, that is Genic balance theory of sex determination. In this lecture, we will study about Genic balance theory, sex determination in Drosophila, sex determination in plants, that is liverwort, then melandrium and maize. So first we'll see Genic balance theory of sex determination. Drosophila, that is fruit fly, possesses three pairs of autosomes and one pair of sex chromosomes in each diploid cell. The sex chromosomes are XX and XY, but the mode of sex determination is quite different. Thus, Drosophila possesses 8 chromosomes and haploid number of chromosomes as 4. 3 pairs of autosomes and 2 sex chromosomes. The sex chromosomes are X and Y. The X chromosome is long and rod-like, while the Y chromosome is hooked, and unlike human, the Y chromosome does not play any role in sex determination of a fruit fly. Every individual fruit fly carries the gene for maleness and femaleness, and the proportion of autosome to X chromosome in an individual determine its sex. Kelvin B. Bridges shows that the female determiner is located on the X chromosome with strength of 1.5 and the male determiner on the autosome with a strength of 1.0 for the autosomal genome. A fruit fly with the chromosome complement AAXX, where A is haploid set of autosome with a male determining value of 1, has the ratio of male. The female determiners as 2:3. Since uh, 3 to 2, the fly will be female. Here 3 is greater than 2. A fly with AAXY chromosome combination will have male. The female determiner as 2:1.2. Since 2 is greater than 1.5, the fly will be a male. This is what genic balance theory. Then here in this chart we can see this particular genic balance theory. Here gametes AX AY, then that produce particular gametes, that's super female, then female. Super female has three set of three chromosome and diploid set of autosomes. Then here female, that 2 X chromosome, 1 Y chromosome, 2 autosomes. Then diploid autosome in male, 1 X chromosome and diploid autosome. Then when this the terms of Y chromosome plus diploid autosome, that fly will die. And the terms of 3X chromosome, that super female also get die. This is what gametes. So remaining gametes get fused with each other and their formation of non-disjunction and their formation of male and female organism of fruit fly. Then here we can see this particular chart showing uh, expression in Drosophila. Thus AX AAXX 2.3, that could express female. Then AAXY, that produce male, that ratio is 2:1.2. 1.2. Then AAXX intersex AAX male, AA triple X super female, that meta female, that low viability. Then triple AXY super male, meta male. Then 4A and 4X tetraploid female. Then double A and XXY, that female. triple A and triple X triploid female. So alternately we can determine the sex of fly by finding the X or A ratio, that number of X chromosome versus number of autosome sites in this table. That is super female having 0.1 ratio, less than 0.1 ratio, female has 0.1 super female has 1.0, less than 1.0 ratio, female has 1.0 ratio, intersex has 1.0 to 0.5, and male has zero point 0.5. Then super male has less than that great more than 0.5. This is the ratio of particular sex determination in Drosophila. Then ratio of sex chromosome and autosomes. Then next is sex determination in plants. Then and most of the angiosperm plant bisexuality, both male and female in one, is more common than unisexuality, only male or female. But unisexuality is more advanced than bisexuality. The plants bearing unisexual flower may be monoecious if the male and female flower present on the same plant, that is maize, coconut, or dioecious, the male and female flower produced on different plant. For example, papaya. Here we can see these different plants. The plant of papaya, that is dioecious plant. Then sex determination in dioecious plants. This sex that heterothallic liverwort, that Mertensia, brown algae, ectocarpus, that UV mode of sex determination. The male have very small V chromosome along with the autosome. The female thalli possess one U and no V chromosome, that is AU. So this is UV mode of sex determination. As stated above, the dioecious plants are those in which the male and female sex organs, Antheridia and Archegonia in lower plants, and male, the female flower and phanerogams are produced on different plants. So that heterogametic male, heterothallic liverwort, that is Mertensia and brown algae ectocarpus, mode of sex determination. Then mechanism Melandrium album. Melandrium, the saline latifolia XX XY pattern of sex determination. Male is heterogametic and having AAXY, and female homogametic having AAXX chromosome. The Y chromosome is largest, having gene formatting maleness, that gene forming maleness, and suppression of femaleness. So this region first, second, and third do not have homogeneous region on X, that is differential portion of Y. V is differential portion of X chromosome, and region 4 is homologous and the chromosome and this particular Y chromosome. This that homologous and linked to the Y chromosome. Then this, here we can see X and Y chromosome of Melandrium. This particular segment, that segment first, second, third, fourth, then 5 and 4 and 5 segment on this particular chromosome of Melandrium. This first is female suppressor region, second is male promoter region, third is male fertility region, 4 is pairing region, after differential region, 5 is differential region of this particular chromosome, differential region of X. Then next is in female, these are the different region. Then next sex determination in monoecious plant. In monoecious plants are those that produce male and female flower on the same plant. Some plants produce male and bisexual flower on the same plant are then termed as andromonoecious, while some produce female and bisexual flower on the same plant they are as gynomonoecious. Then sex determination in males. Then when another type of monoecious plant are most produce male and female and bisexual flower on the same plant. These are called as trimonoecious. So first we'll see sex determination in giametes. Giametes is monoecious plant that produce only unisexual flower in separate male called tassel and the female called ear inflorescence. Tassel is produced terminally and ear is axillary. Unisexuality in maize the plant is selective as stamen in ear and pistil in tassel is eliminated during the development. So here we can see tassel which terminally produce and that ear is produce axillary which produce in axils. Regarding to this, the two mutants were studied to show how the male and female flowers are formed. The anther ear N1 dot D1, D2, D3, D5, and this mutant, the female ear have stamen as it prevent abortion of it, but these are recessive. Benson in 1995 showed that these mutants D1, D3 and AN1 have genes code for endogenous biosynthesis of plant hormone, the gibberellic acid. Gibberellic acid signaling is thought to be preventing the depression of stamen in the D8 mutant. Thus, the genes provide direct evidence for endogenous production of GA, that is gibberellic acid, having feminizing role in sex determination. Then here we can see this mechanism that male that bisexual plants that here pistil abortion takes place, then stamens get arrested, and here we can see D1, 3, 5, D8, then here AN1, SK1, PI, P2. Then next tassel seed and sickle mutant. Tassel seed 1 and tassel seed 2 mutant of maize feminized male floret in tassel. The genes have found to be produce alcohol dehydrogenase and steroid dehydrogenase which involve in pistil abortion. This TS1 and TS2 genes affects only primary sex character, while TS4 and TS6 affect secondary sex characteristic as well. The TS6 mutant, for example, feminizes the tassel and disrupt branching and inflorescence. Then in SK plant, the pistil and stamen of ear floret are aborted, although secondary character are remain same for female, indicating that SK1 necessary for the formation of functional pistil in the upper ear floret. Then in this way we have seen this sex determination in different plant. Now we'll see the remaining part in this genic balance theory, that is gynetomorph. This gynetomorph, there are sometimes when in unfertilized egg of Drosophila, the first mitotic division of the zygote or later mitotic division of embryo may have irregularity. This would produce flies which are half or part female or half or part male or at time intersex. Such flies are known as gynanders or gynandromorph. This we'll see in next lecture. And uh, in this lecture we have studied only this genic balance theory, then sex determination in dioecious plant, and sex determination in monoecious plant. Thank you.